Well, so again, thank you very much. So what I'm going to cover today, oh, this is working. Oh, there we go. Thank you. So basically, my lab has very broad interest in cardiovascular regeneration, so anything related to heart and vascular function. And when we're talking about mitochondria and metabolism, we really need to think about that some of these heart failure and heart diseases are really driven by the metabolism, right? And how can we bridge this and how can make these things more relevant and understand better what we're doing? Another main focus of the lab is diabetes. So again, kind of merging these things and seeing how one thing affects the other. And in the more engineering side of the lab, we do in vitro models of health and diseases, which we call organs on chip, and 3D engineered tissues. So I'm going to give you just a little bit of flavor of the different projects that we have in the lab. It's not comprehensive. We've been doing a lot. But so one of the main things we're doing in terms of translational work and regeneration relates to therapeutic vascularization. So one of the first things that go, I guess, when you get certain diseases, and diabetes is one of them, is the vessel. So vessel function is really impacted by diabetes, and we all know that. So in my lab, what we're trying to do is, if we're trying to create these therapies to treat the heart or transplant islets, to treat some of these diseases, and so to have the body to be able to restate the function, right? And we don't need to depend on meds anymore. We need these cells to survive. And the biggest issue when you transplant these cells is survival, right? If the vasculature is not there, they're not going to be able to survive. So one of the things we do is basically recycle blood vessels from tissues we do not need to survive, such as the fat tissue, and we co-transplant these vessels with heart cells or pancreatic islets in different models. And we've shown that these can not only improve the graft size and survival of these cells in different contexts and different diseases, but also can reverse function. So if you see on the bottom left, the black lines were cells transplant animals that received cells with the microvessels, and you can see the function of the heart, it's increasing, so it's going up. And same thing for diabetes. If you look at the black line, which is the combination treatment with the microvessels, you see a significant decrease in blood glucose in these animals that had high blood glucose before. And this is really hard to do because endothelial cells do not like to be transplanted. We've tried, and the field has tried, putting endothelial cells in all these different organs, and it's very hard to get them to survive. So now we're trying to figure out what's the secret sauce in these vessels that we take from fat that make them so special that they stick around and that they provide a lot of survival. And then the other part of the lab is focused on, as I said, organs on a chip. So these are not really organs, right? They're basically the functional unit of the organ that we can use to study the organ. In this case, we're talking about a heart on a chip. So you can see here this beating piece of tissue that is hanging into this rod. And I think you guys are going to have some more talks that are going to show you some of these models, where we've mixed cardiomyocytes, which are the active cells that pump help with the pump function in the heart, with fibroblasts to create these tissues in vitro. So these are human cells. So these make these models more human relevant, because we also know that, sometimes the studies we do in animals can't really translate to humans. And in the heart, that's particularly true. There's a lot of drugs that are recalled from the market because they have toxicity that's specific to human cells, and etc. So these models help us to have a live, real-time assessment of function and how certain drugs or different diseases affect contraction and function of the heart cells. So we've been using these to do a number of disease models in vitro. So some master students in the lab generated this model of cardiac fibrosis on a chip, where we could study all these different parameters, so transcriptome, the extracellular vesicles, the position of extracellular matrix, test drugs, and etc. And more recently, a PhD student in the lab, Omar Morad, has shown that by treating these this fibrotic tissues in vitro with antisenescence drugs, we could improve function. Now, we've tried to treat these tissues with different drugs, and we get improvement in some parameters, but rarely we get improvement in function. And this was the one case, only with the senescence, that the senolytics, where we could see this improvement in function. So we're really excited about this and trying to learn more about mechanism. When we've also been able to show that we can create complex arrhythmias in vitro. So one of the issues is that, it's really hard to study arrhythmias in vitro. And in this study, we've shown that if you focus on the optical mapping part, the top in the control, the black line shows, that when they beat, they contract and they have this front of migration of activation. So they're all activating synchronously. But in the bottom is our arrhythmia model. You can see all these different arrows pointing to the way the wavefront of activation is working in this cardiomyocytes. And what happens is they can contract together, which means they can't generate function or pumping function. So this is all really nice, but now, and this is a little bit more relevant to what Saf is going to talk to you soon, we've been dabbing into creating perfused vasculatures in vitro. And this is important because, we've always studied endothelial cells in a plate, which has been extremely helpful for us to understand a lot about the endothelial function, but we also know that perfusion is a key parameter for the endothelial cells. So by assembling endothelial cells into these devices in specific gels and providing flow, you see that the endothelial cells assemble with time into this vascular network that you can see by red now that they can be perfused. And this allows us to study a whole number of different things within these tissues. And Kayla, who was a master student in the lab, published this paper on how we showed that if you basically dope these healthy endothelial cells shown in green with the endothelial cells shown in red that have a clinically relevant mutation for brain arteriovenous malformations, which are these enlarged vessels in the brain that often have to be removed surgically but have no real treatment for. They form these more dilated areas in the vascular tree. So with that, I wanted to thank everyone in the lab for doing this work and you for the opportunity to present. And I guess we're going to move straight into our great speakers of today. So we have 4 speakers. These are trainees who received funding from MITO2i's, which we're really thankful for, and allowed, in particular in my lab, us to kind of dab a little bit more into some of the metabolism work. So our first speaker is Safwat from my lab. In the Department of Biomaterials and Biomedical Engineering, and he's going to talk to us about elucidating the role of mitochondrial dysfunction in islet endothelial cells during type 1 diabetes.
Here. Okay. Okay, all right. Okay, hello everyone. My name is Safwat. I'm a PhD student in the lab of Dr. Sara Vasconcelos, and today I'm going to be presenting to my research about elucidating the role of mitochondrial dysfunction in islet endothelial cells during type 1 diabetes. So just a little bit of background. So endothelial cells are the cells that make up the vasculature, and they're very imperative for the proper functioning of the insulin-secreting beta cells. Specifically, apart from acting as a barrier between the blood and the tissue, they also have a very intricate crosstalk with the beta cells that is important for its function. During type 1 diabetes, so during type 1 diabetes, the endothelial cells become dysfunctional, and that causes a lot of problems. For example, this allows the infiltration of these leukocytes that end up destroying the beta cells and also affecting the crosstalk with these beta cells. And we know that from a large body of evidence that endothelial cell dysfunction is actually mediated through mitochondrial dysfunction. Therefore, in our research, we hypothesized that this diabetic microenvironment, which includes the inflammation and the hyperglycemia, can induce endothelial cell dysfunction through mitochondrial dysfunction. And this leads to the exacerbation of the type 1 diabetic status indirectly through the leukocyte infiltration and directly through the breakdown of this crosstalk. So if we can basically target this mitochondrial dysfunction, then we'll be able to treat endothelial cell dysfunction and hopefully alleviate the disease burden of type 1 diabetes. So in our research, we have analyzed single-cell RNA sequencing data from islets in a type 1 diabetic mouse model. Specifically, we're showing the single-cell data of endothelial cells. And essentially, the axes are basically a summary of all the entire transcriptome into 2 dimensions, and each dot is a cell. So as you can see that the endothelial cells cluster into 2 separate clusters of diabetic and non-diabetic endothelial cells. And this is because the diabetic endothelial cells have these leukocytes, and the non-diabetic don't have these leukocytes. And you can see in the diabetic endothelial cells that there is a lot of cells also characterized as inflammatory. And within this, we also have different time points to the diabetic progression. And you can see as the diabetes progresses, we have a higher percentage of these inflammatory endothelial cells. So if we can find out what is different between the diabetic and non-diabetic endothelial cells, we can be able to identify targets to prevent this kind of dysfunction. So we looked at whether the genes that are different between diabetic and non-diabetic endothelial cells, and we looked at whether the processes that are aberrated. And among many other terms, we found that a lot of the top terms that were aberrated were actually related to mitochondrial function, suggesting that there's mitochondrial dysfunction in the diabetic endothelial cells, such as oxidative phosphorylation, electron transport chain, etc. And so if we can identify the genes that actually classify non-diabetic and diabetic endothelial cells, we can identify targets for mitochondrial function treatment. For example, one of the top targets was MCL1, which is involved in mitochondrial calcium homeostasis. So once we have identified these targets, we can actually try to assess them in our 3D models and in vitro models that Dr. Vasconcelos mentioned. So we can create a physiologically relevant diabetic model using pancreatic microvascular endothelial cells. And we have shown in a preprint that endothelial cells are very, very heterogeneous, and islet endothelial cells specifically have a very tissue-specific characteristic. Therefore, by creating a diabetic microenvironment, by inducing inflammation through cytokines and high glucose treatment, we can assess whether it is actually increasing mitochondrial dysfunction and endothelial cell dysfunction. And then we can use this, actually use small molecule treatments to see if we can reduce this mitochondrial dysfunction and endothelial cell dysfunction, and hopefully identify some treatments that we can hopefully port to type 1 diabetes treatments. So that's a very, very quick summary of the research plan. And of course, as through all research, we should be very aware that our research should be patient-focused. So for that, we hope to that once we have found these findings, we hope to author articles written in lay summary, so we can publish them on our lab website so patients can actually go in and read them and understand what this research means to them. And we also hope to present these research at patient engagement seminars, such as those held by Diabetes Action Canada. Finally, the benefits of this is kind of clear, where you can see that if we can find treatments for mitochondrial dysfunction, we'll be able to identify treatments for type 1 diabetes, as well as other autoimmune-related diseases that include endothelial cell dysfunction, such as ischemic bowel disease. So with that, I would like to thank MITO2i for giving me this opportunity to present my research and also for funding this project, as well as my lab members and some other funding agencies as well. So thank you very much, and I hope you guys enjoy it.
Thanks, everyone. We won't have time for questions now, but I encourage you to talk to the speakers during lunch. So our next speaker of today is, Yuanye from Amy Ramsey's lab in the Department of Pharmacology and Toxicology, and her talk is going to be on beta-hydroxybutyrate, improving behaviors and mitochondrial function in a mouse model of grin disorder.
Hi, everyone. My name is Phoebe. I'm a PhD student in Dr. Amy Ramsey's lab who just gave a talk on the same project, basically. So hopefully it'll make my very condensed short talk easier to understand and follow. So basically, our lab studies grin disorder, which is a neurodevelopmental condition caused by rare genetic mutations in one of the grin genes that encodes NMD receptor, which is very important for synaptic plasticity, therefore learning and memory, but it also mediates excitotoxicity. All different classes of this grin variants has been identified. That leads to over or underactivation of this NMD receptor function, or sometimes it's a very complex condition. Therefore, it makes the conventional pharmacological treatments directly targeting NMD receptors very challenging. And the field has been trying to stratify and characterize all the variants, but at the same time, we've been thinking, are there some convergent downstream pathways that could be targeted and actually improve the symptoms? So one of the things we see is the patients with grin disorder suffer a broad spectrum of phenotypes, and many of those are also seen with those that has mitochondrial diseases, such as the developmental delay, seizures, movement disorders, and so on. And there is, of course, a lot of accumulating evidence associating NMD receptor with mitochondrial dysfunction that I don't want to go deep into. We also saw this high energy demand, but reduced glucose uptake in our GRIN1 knockdown mouse model really made us to ask, can alternative energy source such as ketone bodies be a potential treatment for grin disorder? So to test that, we put our mice on exogenous ketone bodies by directly putting BHB into their drinking water or put them on a ketogenic diet, as mentioned, Amy already introduced. Parallel to that, we also wanted to directly looking at whether the BHB could improve mitochondrial function. Therefore, we cultured some cells from the cortex of the GRIN1 knockdown mice and then give them some BHB, and 1 day later we did the metal stress test using the seahorse analyzer to directly looking at mitochondrial function. So for the behavioral part, although we see both BHB and keto diet could improve their hyperlocomotion in the GRIN1 knockdown mice, however, half of the GRIN1 knockdown mice specifically were meeting endpoint that received ketogenic diet. On the contrary, the BHB supplementation was very well tolerated and achieved similar improvements. For the mitochondrial function part, we did see kind of surprisingly, but also as we expected, there is overall increased oxygen consumption rate in the GRIN1 knockdown mice. They had increased basal respiration and increased ATP production, which both seem to be improved or reduced by the 4 millimolar BHB we applied before we did the test. Of course, this is just preliminary data that we want to see if we can repeat, but this is very exciting. Moving forward, we do want to go more patient engaging, so we've generated this patient variant mouse models that carries the exact same missense heterozygous mutations that we're seeing in the patients. We want to see whether those bioenergetic dysfunctions were also seen in these models and whether BHB could improve those outcomes. And also our lab works very closely with CureGRIN, which is a patient organized foundation that connects the families with clinicians and research that are studying grin disorder. And actually the main reason we're studying this is because this is so whether we can targeting downstream pathways, one of the top 10 questions that the patients wanted to be answered by the researchers. I'll just end my short talk there. That's all. And I want to thank everyone in my lab, my committee member, Dr. Anna Andreazza and Dr. Rob Bonning. And of course, thanks MITO2i for giving me the funding and the opportunity to talk. Thank you.
Thank you very much. That was great. And next speaker is Kassandra Zachos from Ana Andreazza's lab in the Department of Pharmacology and Toxicology. And she's going to talk to us about the implications of dietary intervention on mitochondrial dysfunction in type 2 diabetes.
Okay, hi everybody. So today I'll be talking about my project that was funded by the MITO2i scholarships, and it is implications of dietary interventions of mitochondrial dysfunction in type 2 diabetes. I don't need to go into details as to what type 2 diabetes is. We've had such a great introduction already, but we do know that there's disrupted glucose homeostasis. Type 2 diabetes is a global problem. It accounts for over 90% of the diabetes cases in adults. There is a pretty dramatic reduced life expectancy of these individuals, and there is a very substantial healthcare cost associated with it. It is an incredibly complex illness. It encompasses environment, diet, genetics, lifestyle, and we know that mitochondrial dysfunction is involved. How or why can vary. It's very personalized and heterogeneous illness, but ultimately it can make the illness and exacerbate individual symptoms with systemic consequences. Our objective was to gain a further understanding of mitochondrial dysfunction in diabetes and assess dietary interventions to see how they modulated mitochondrial dysfunction. And the way that we've gone about examining so is looking at 2 mitochondrial blood-based biomarkers that we can find circulating through our periphery. The first is elevated lactate, which we briefly heard about this morning. Lactate is traditionally known to be elevated due to a metabolic shift where the mitochondria is not able to function adequately to produce sufficient ATP, and we have to rely on glycolysis and other measures to produce ATP and energy. This can lead up to a buildup of lactate that, while is incredibly important in homeostasis, at elevated levels can have systemic consequences, including increased gluconeogenesis, increased lipid metabolism, increased cholesterol production, and increased glutamine metabolism. On the other hand, mitochondrial DNA is incredibly sensitive. It's this plasmid circular shape that is prone to fragmentation. When we have mitochondrial damage or mtDNA fragmentation, it can be released and accumulate in our periphery, and that is called circulating cell-free mitochondrial DNA, or CCF mtDNA, as you'll see on my slides. This can lead to chronic low-grade inflammation and increased expression of pro-inflammatory genes and inflammasome activation. So we measured these biomarkers in individuals that underwent 2 different dietary interventions. The first is a high-fiber cereal diet. So think the brown option for carbohydrates, brown rice, brown breads, that sort of idea. And then the second diet has low glycemic index diet. So the carbohydrates are focused on things like quinoa, barley, ultimately things that don't cause a dramatic spike in our blood sugar. The rest of the diet remain the same between individuals. And we hypothesized that subjects following this low glycemic index diet will have lower CCF mtDNA levels and lactate levels. Before I jump into some of the outcomes, patient engagement is crucial. The study obviously could not have taken place without the willing participation and enrollment of over 150 individuals. The Jenkins lab, which I worked with closely for this work, recruits tons of individuals, and there's been trials, dietary intervention trials in diabetes going there for decades. Patients were followed up throughout the 6 months of this trial. The diet intervention was 24 weeks. They were followed up monthly in clinic and had 3 biosample collections throughout the period of the trial. There is a discrepancy in some instances between model organisms, so it's really important that while we investigate these mechanisms in other model organisms, that we include human investigation and trials as well. Diabetes is an incredibly complex disorder, and I think we need to work with participants in order to better understand and develop a personalized approach. It can help us generate proactive medicine alongside pharmaceutical therapeutic production, which is more reactive medicine. And the primary goal is to provide meaningful research to these participants. And without their involvement, I don't think we can do that. So uniquely, we actually didn't see any significant changes in lactate or CCF mtDNA over the 24 weeks. While that may seem like a negative result, in the interest of biomarker development, it actually could be pretty positive and can mean that these are more robust biomarkers than we may give them credit for. What we did see was that the change in CCF mtDNA from weeks 0 to 24 correlated with cholesterol, LDL, and systolic blood pressure in only the low glycemic index diet. We also investigated CRP, or C-reactive protein, which is an acute and inflammatory marker. And in the original study that they conducted with these diets, the CRP actually went down in both dietary interventions. And then in our further analysis with the biomarkers, we noticed that CRP was correlated to cholesterol only in the high cereal fiber diet and was correlated with BMI in the low glycemic index diet. So this definitely warrants further investigation, but it provides a pretty unique insight into how different interventions can ultimately affect mitochondrial dysfunction. Thank you to Dr. David Jenkins and Dr. Andreazza, the MITO2i team, for allowing me to speak here today, and my lab members.
Thank you. That was great. Apologize because I'm probably gonna butcher the name. So our next speaker is Dana Soufi Sabbagh from Ana Andreazza and Peter Carlin's lab in the Department of Pharmacology and Toxicology, and she's going to talk to us about using cerebral organoids to study mitochondrial dysfunction in bipolar disorders. Thank you.
Hello, everyone. My name is Dana, and I'm also a PhD student at Dr. Ana Andreazza's lab. And today I'll be giving you a little bit of an overview about using cerebral organoids to study mitochondrial dysfunction and bipolar disorder. So bipolar disorder is a debilitating disease. It affects millions globally. It's characterized by severe mood swings and energy imbalances in patients. Bipolar disorder is pretty variable amongst patients, and it's pretty complex to model as well. There are two current implications in the literature on bipolar disorder. One, where it is being increasingly classified as a mitochondrial disease, and this is because of dysfunctional mitochondria, dysregulated cellular energy metabolism, where we see varying levels of lactate and ATP, et cetera. The other arm is this chronic underlying inflammation we see in patients with bipolar disorder. And this is usually stemming from the increased levels of reactive oxygen species, where the NLRP3 inflammasome, a redox sensor, senses this elevated ROS and activates this pro-inflammatory cascade. So today I'm going to go over a small research plan about what we're planning to do in terms of investigating bipolar disorder using mitochondrial dysfunction, as well as this underlying inflammation through the use of cerebral organoids. For those of you who are not too familiar with the cerebral organoid field, they are three-dimensional tissue structures, making a very simple model of the fetal brain. They are comprised of neural progenitor cells, neurons, and astrocytes. At the top right is a little image of what an organoid, a cryosectioned organoid at a 1 month time point, where they are mainly staining for SOX2 positive neural progenitor cells. And these organoids actually have a self-patterning organizational structure, where at 1 month, they are mainly comprised of neural progenitor cells. And as they mature to 4, 5, and 6 months, these NPCs further differentiate into neurons and astrocytes. Now, what we've done is we've collected blood from patients with bipolar disorder and control patients, generated iPSCs from this blood, and then differentiated these iPSCs into cerebral organoids. We then take these organoids and run a number of mitochondrial assessments, where we look at their mitochondrial membrane potential, difference between mitochondrial mass, polarized healthy mitochondria, circulating cell-free mitochondrial DNA release. And we also look at the levels of NLRP3 inflammasome activation in the BD and control organoids to get a sense of what's happening between the two groups. Now, as important as it is to understand the underlying mechanisms of bipolar disorder, it's also just as crucial to make sure our research is actually benefiting those we intend to. And this slide highlights the framework of our patient engagement strategies, where we begin looking at patient perspectives and patient experiences from our study design all until the feedback integration, because it's not just important for us to have statistically significant results, it's also important to have these results be scientifically important to directly benefit the patients later on. And a little bit of some outcomes from our study so far, just a few data points to highlight. We did a metabolomics assay on the cerebral organoids and found some interesting results where we saw decreased levels of glucose 6-phosphate in the BD group organoids in comparison to the control, indicating some sort of glucose buildup in the bipolar organoids. We also saw decreased levels of ATP in the bipolar organoids, and this was further confirmed by secondary assays in our lab. We then went on and at the top left image stained for mitotracker green, which looks at mitochondrial mass in the organoids, and we found no differences between the control and the BD group. However, when we stained for mitotracker red, which is the bottom left image, we actually found a reduction in the BD group, where mitotracker red stains for live, polarized, healthy mitochondria. So this tells us the BD organoids seem to have similar levels of mitochondria as the controls. However, not all the mitochondria are as functional as the control group. In terms of the inflammatory side, we assessed the NLRP3 activation at baseline in the control and BD groups and saw no significant difference between the two groups. However, after exposing the organoids to LPS and nigerisin, which are NLRP3 inflammasome activators in vitro, we actually found the BD groups was twice as likely to be activated, making them more prone to this inflammatory cascade we typically see in patients with bipolar disorder. So I would like to thank our team at the Andreazza lab, the Machado lab, the Carlin labs, and Liliana's Atisano's lab. Thank you very much.